Okay, it's great to be here. You know, the HFSP has supported my research from the beginning. When I first became a group leader in Heidelberg, I had an HFSP program grant with Tom Pollard. And so I thought I'd tell you today about a story that illustrates two of the key values of HFSP. One, young scientists should travel abroad. And two, discoveries often come from studies that cross disciplines. So I want to start by considering this giant sequoia tree, which you find in the redwood forest near San Francisco. Now, this tree's lived 100 years, or hundreds of years, and to do so, it had to constantly make correct and accurate decisions in the face of thermodynamic shocks, such as temperature, nutrients, salt, pH, and water. Now, this process requires the coordinated response of trillions of cells, right? This sequoia on its own consists of trillions of cells, and to put that number in context, one of the galaxies you just heard of in a previous talk, there's only 100 billion stars in the Milky Way. So each tree is far more complex than the number of stars in any one of these galaxies. We think that most of these cells are regulating their biochemistry through the formation of compartments, and that the way they regulate these compartments is key to how they respond to thermodynamic shocks. These compartments come in many flavors, such as those bound with membranes, which Randy Sheckman did his key work on, and those not bound in membranes, the nuclear pore. But we think that it's the way that these compartments respond to these different thermodynamic shocks is key to the way cells regulate themselves. But a cell itself is unbelievably complex. Just to put that in context, if we take a cell and look at a compartment, so here's one compartment, so in red, and ask how many proteins are in one compartment, we come up with some amazing numbers, like 1 million proteins in one of these compartments. And if we ask how many proteins are in a cell, we come up with about 5 billion, which is roughly the population of the world. So you can think about, the formation of a compartment as sort of about 1 million molecules coming together out of 5 billion as roughly, say, building a small city. People would come from all over the world, they would build a city, and then they would go back again when it's finished. But the cell is an even more bizarre place than that, because if you scale up proteins to be people, but you maintain the same density as if they're proteins, we'd actually all fit in the Great Salt Lake in Utah, right? We'd be about arm to arm apart, tumbling past each other about 3 square kilometers a second. And this extreme physical-chemical environment is the environment in which cells are constantly making decisions about how to respond to these thermodynamic shocks. The question of how cells build these compartments is something I was lucky enough to work on already as a PhD student. I came up to do my PhD at the LMB in Cambridge. I arrived for my interview, and I could see I wasn't doing very well. It was all on DNA and that sort of thing, and I knew nothing about DNA because I'd done my undergraduate at UCL, where they told us more about cell biology, until I stumbled into an office with John White, who showed me these amazing movies of C. elegans embryos. And I just fell in love with his cell division. And I've worked on that for the last 40 years. You can see the 2 pronuclei coming together and cell division. So that was a lucky break, being fascinated with how this thing builds its internal compartments. So the story today starts with the fact that to understand a problem like this, it helps if you can work in a cross-disciplinary environment. And this cross-disciplinary environment came after the wall came down between East and West Germany. I had the luck to be part of a founding team to start a new institute in Dresden in the old East. But the luck I had was that Max Planck decided to build a second institute, the Physics of Complex Systems, and hire Frank Ulicker. And together we settled down to think, how can we use physics to help us understand this problem of how a cell assembles? Now, the second feature of the HFSP is young scientists benefit from travel abroad. And young scientist in this case is Cliff Brangwijn, who I met when I was teaching students at a physiology course in Woods Hole. He was a physicist who'd been trained by Dave Waits. And when I met him, I persuaded him that a postdoc in Dresden, which is definitely not high on his list, as a Harvard graduate student, I think he was imagining going to Stanford or some of the other big places in America. But he decided he'd be very interested in coming in to this cross-disciplinary environment, which he did, and started to think about how physics could help us understand this C. elegans embryo. And the problem he decided to focus on was this one of how these type of compartments called P granules segregate to one end of the embryo prior to division. They'd be discovered by Susan Strohm and Bill Wood when I was doing my PhD, an amazing example of intracellular segregation where they end up at one end of the cell, where they go on to make sure that the worm that grows up has babies. Now, of course, when you look at that, you think, that looks like flow transport. And actually, that's what Cliff was interested in. But I dug up a slide, which I probably haven't shown for 15 years, which is Cliff comes into my office and he says, Tony, we've got a problem. Because as a physicist, he actually knew how to be quantitative, right? And at that time, as biologists, we didn't really know anything about quantification. And he said, the number going one way is the same as the number going the other way. So it can't be transport. But I was thinking of something much more interesting, which is these P granules seem to behave like liquids. Look at this. You can see them dripping, sort of like honey off your spoon in the morning. And you can see them fusing, kissing, coming apart, and fusing, just like a liquid drop. Now, why would you get so excited about this? It's because liquids have all sorts of interesting properties, such as viscosity or surface tension. And we've begun to think, could these properties affect biochemistry? And the most one we focused on was this concept of phase separation. You're very familiar with phase separation, for instance, from clouds. Anyone who's ever laid on their back on a grassy and looked in the sky and see clouds form, see they come just out of nothing. And that's because the water vapor evaporates, and as the temperature gets cooler and cooler and cooler, and suddenly, when it reaches the dew point, it condenses. And this illustrates a phenomenon of phase separation, which is change comes all at once. And physicists tend to draw a phase diagram to illustrate this. In this case, we might have temperature. Here I've done protein concentration. Anything underneath this line, you would get some phase separation, anything above. So if we lower the temperature, we move into a regime where they now form. So that's okay for macroscopic physics, right? But the question we asked ourselves is that sort of physics actually useful in thinking about the cell? And this took us about a decade to do the experiment of asking whether you can see phase separation by building a stage that allows very precise temperature and spatial control of temperature. And this allowed us to do the following experiment, which is here we have some P granules, and we're going to cycle the temperature up and down. And you can see them forming, dissolving, forming, dissolving, dissolving, forming. And from that, we could draw a phase diagram. So that was sort of prima facie evidence that C. elegans P granules are liquids and formed by phase separation. So then we can ask the question is, do cells use phase separation to regulate biochemistry? And I've talked about this temperature response. And the whole point about a phase separation, it provides a unique physicochemical mechanism where very small changes in parameters such as temperature or salt or pH lead to enormous changes in state. And the best examples have come from plants, which is why I showed the tree at the beginning. You know, when we are subject to thermodynamic shocks, we can move out of the way, but plants have to just sit there and take it. So not surprisingly, they've used phase separation extensively to rapidly respond to thermodynamic shocks. And we know how important this is. This just shows the change in bloom date with time, and we know that global warming is leading to changes. And so, but how is it that plants are actually recognizing and regulating response to temperature? And this is one experiment I like to show from Phil Wigger. It was about Arabidopsis. And he asked the question, how do rhodopsids know how to flower at a very precise temperature? And he showed that this repressor complex, ELF3, condenses out at a certain temperature and takes it out of action, allowing plants to start flowering. So you can think about it this way. There's a repressor complex that's active and represses flowering. And then after phase separation, it forms this depot, takes it out of action, and inactivates it. And for those in the audience who don't think about these things, you could think about a set of people who have to go to work, and if you force them into a confined space where they can't go to work, then they're unavailable. They're a sort of inactive depot and can no longer contribute to the process. And there are lots of other beautiful examples of that in the literature. This is one of my old favorites as well, which is how do cells respond so rapidly to water influx? And this paper talked about a kinase which phase separates and rapidly activates to change the receptors. So those experiments have shown you how cells can use phase separation as a very rapid way to respond to thermodynamic changes. But I want to just finish on one third aspect of the core of the ATFSP, which is that basic science provides the knowledge that drives innovation. And that comes back to the question about can we think about phase separation and help it in drug discovery? Now, the core problem in drug discovery is that only 15% of the genome is druggable. That's a massive issue we all face. We also know that governments are getting less and less keen on paying the enormous costs it takes to develop drugs, suggesting we need new ways to get at this problem. Now, one of the key proteins that is undruggable is beta-catenin. It's probably involved in 100% of colon cancers, maybe 90%, probably 15% of all cancers. But it's been undruggable because of a lack of an enzymatic site. It's a frat protein-protein interface. So we went back to think about this experiment. If you can take a protein out of action, cells can do it by phase separation in this plant example. Remember I said when the repressor complex goes, could we do that in drug discovery? In other words, could we take beta-catenin and drive it into a depot and use that as a way to inhibit its activity? Right, so this would be like taking our beta-catenin out of the factory, and so it's no longer available for work. And indeed, we've been successful in that, right? So here's a healthy cell, and here's a drug from produced by a biotech company I started together with Rick Young. And that, in healthy cells, it doesn't form any depot, but in disease cells, it does form this depot that takes it out of action. And sort of amazingly, the concentration at which it forms a depot is pretty similar to the concentration at which it kills cancer cells. So that's just about to go into patients, so we're hoping it's obviously going to be successful in beta-catenin-driven cancers. And just for those of you who think about the physical chemistry of this problem, it's a very interesting example of something that physical chemists call polyphasic linkage. So here is temperature and protein concentration, and here is the phase diagram, say, of beta-catenin. Now, this would be the concentration of the wild type, and in the mutant, in cancer, it's been upregulated in its concentration. So what these small molecules do is they shift the phase diagram into an area where the mutant now phase separates and the wild type does not. As I said, a process known as polyphasic linkage. And that has two great factors. One is that it allows the mutant to start phase separating, but also it leaves enough protein that is soluble that can actually drive the wild-type normal functions of beta-catenin. So I just wanted to finish. Pavel asked me just to make one point here, which is this, I've told you today, illustrates, I think, the three key values of the ATFSP. First, this is a pretty obscure observation, right? What happens to a pea granule in a nematode, right, that forms a liquid? And that was driven by movement of talent, right? It happened as Cliff decided to go from Harvard to Dresden into an interdisciplinary environment that we set up. It generated a basic knowledge base, right? It wasn't driven by the idea of solving any problem, but in fact, it's allowed companies to look and think how they can change drug discovery, but of course, also generates knowledge of philosophers or the general public to think about these kind of problems. And I'd like to thank you for your attention.